Hi there guys. Today what we're looking at is the chemical structure and function of proteins within the body. So proteins make up a huge variety of animal and plant material, for example meat, fish, chicken, eggs, all things that we eat in our diet, and without proteins our bodies could not survive. Proteins are the major structural materials of animal tissue, and they're also involved in things like maintenance and regulation of life. So amino acids are the building blocks of proteins, and they are linked together using a condensation polymerization reaction. So condensation, when you join two molecules together with the loss of water. And we've seen this previously when we've looked at esters and condensation or esterification reactions. Amino acids are the building blocks from which proteins are formed, and they're molecules which contain two functional groups, an amino group and a carboxyl group. So for example, if I was to draw an amino acid, I would have my carboxyl group at one end, C double bond O OH, and an amino group at the other end, NH. And amino acids have an R group in them, and then this R group can contain different elements, carbon or hydrogen or nitrogen or oxygen, and it is the R group which differentiates one amino acid from the other. So the most simple amino acid has its R group as just one hydrogen, and that R group changes for every individual amino acid, and there are 20 different amino acids which are used to make the proteins needed in a body. When two amino acids join together, that is called a dipeptide. So two amino acids di, and peptide meaning protein chain. Three amino acids joined together is called a tripeptide, and if we join many amino acids together, that is called a polypeptide, and that is what we call a protein. So the body cannot make all of the amino acids required for protein synthesis, and the amino acids that the body can't make, that we have to get through our diet, is called essential amino acids. So we have to eat those amino acids through eating things like meat or fish or eggs in order to get the amino acids that our body needs to regulate itself. So for example, if I'm looking at this protein here, then I can see that this here is what we call an amide link, where our amino acids have joined together. So this here is one protein, one amino acid, sorry, and this is another amino acid. So this is a dipeptide. If I look at this example on the right, then I can see where I can split my amide links, and I've got my first amino acid here. I've got a second amino acid here, because I can see that that is my carboxyl group and my amide group, carboxyl group, amide group, carboxyl group, amide group. So at the end here, that's my third amino acid. So this is a tripeptide. So as I said before, we've came across condensation polymerization previously when we've talked about the condensation reaction between carboxylic acids and alcohols to make an ester group. Now we're talking about the condensation polymerization of amino acids, so the reaction of a carboxylic acid of one amino acid and the amine group of another amino acid. And I'm going to show what that will look like. So for example, if we use two amino acids, so we've got our carboxyl group and our amine group next to another amino acid, carboxyl group and amine group. I'm using the R here to just simplify my amino acid. That R group could be anything. It could be a long chain, it could be a ring, it could be just a hydrogen by itself, it might be two hydrogens. But these are just general amino acids here. And during condensation polymerization, we are losing a molecule of water. And that molecule of water is lost, a hydrogen from the amine group of one amino acid and a hydroxyl group of the carboxyl group on another amino acid. So I'm losing that molecule of water. And what I'm doing in the process of that is forming a new bond between this nitrogen and this carbon, because that is going to be lost. So I'm taking away one molecule of water. This side of my amino acid has not changed, so I can just copy it out as it was. And now between my nitrogen, it still has a hydrogen on, but I have a new bond to my carbon C double bond O. And the rest of that molecule is exactly the same as it was. So we can call this molecule a dipeptide because it's two amino acids joined together. So this is my new dipeptide. And what I've made is a new functional group. I've taken two functional groups and reacted them together to make a new functional group, which is this NH C double bond O. That is my new functional group, which holds my amino acid molecules together. And we call that functional group an amide link. An amide link, that is what holds together proteins and what holds together amino acids to make long chains of proteins. So proteins are formed from a condensation reaction whereby amino acids join together and a molecule of water is eliminated. The new bond formed in the protein where the amino acids have joined is called a peptide link or an amide link. So I'll highlight that as well. You can use those words interchangeably, peptide link or amide link. Proteins which fulfill different roles in the body are formed by linking together different sequences of amino acids. So depending on what protein that the body wants to form, it will take amino acids and it will link them together in different orders to make different proteins. In our body, proteins have a very specific function. The majority of proteins in the body are used for growth and repair of tissue, but proteins are very large, they're very important and complex molecules in the body, and they have a variety of functions. So the table below shows some of the functions of different proteins in the body. So for example, we've got keratin, which is involved in the protection of hair. We've got actin and myosin, which are involved in the movement of muscles. We have insulin, which is involved in the regulation of glucose. We have amylase, involved in the digestion of carbohydrates. And hemoglobin, which is involved in oxygen transport. So you'll see that some of these proteins are fibrous and some of them are globular. We're going to talk about that in a second. But for the types of proteins that they are, we've got structural proteins, we've got contractile proteins, hormones, enzymes, and proteins which are involved in transport. So there is a variety of jobs that proteins do in the body. So when proteins break up in the body, we call this digestion. So when our bodies digest proteins, we get the amino acids back that we started with. Now this reaction is a hydrolysis reaction, which we've heard before when we've talked about esters and hydrolyzing esters. It is the same reaction where we are taking water molecules and we are breaking apart peptide links to leave the separate amino acid molecules that we started with. So if we look at a dipeptide again, a simple dipeptide, two amino acids joined together. So I've got two amino acids joined together here, and I know this because I can only identify one peptide link in the molecule, one bit in the molecule where I have NH bonded to a C double bond O. So during hydrolysis, we are splitting that peptide link apart. So we're splitting it down the middle to get the two amino acids back that we started with. And we do that by adding water back into the molecule. So if I'm splitting where I've drawn my line here, then the first amino acid is going to be this OH double bond O C H R NH, and when I put water back in, one of the hydrogens from the water adds back onto my amine group to get my amino acid and my complete amino group, which is NH2. I will also make my C double bond O C H R NH2. So this molecule here, except onto my C double bond O, I'm adding an OH from the water molecule. So effectively, this water molecule here I'll do it in green. This water molecule here, one of the H's adds to one of the amino acids and the OH adds to another amino acid. So after I've chopped my peptide link down the middle and separated the amino acids out, the NH from one amino acid gets another H to make a complete amino group, and the C double bond O of another amino acid gets a hydroxyl group OHO to make a complete carboxylic acid group. During digestion in our bodies, enzymes are used to break down the proteins, and these amino acids are transported around the body by blood. So once you've got a protein in your body and your body has broken it down by digestion or hydrolysis, those amino acids are transported around the body in the bloodstream. And condensation reactions build up the amino acids to produce proteins required by the body. So once our body breaks down the amino acids, those amino acids are transported around the body and our body uses those amino acids to make new proteins. There are different types of proteins in the body, and they can be categorized into two main groups. The first group is fibrous proteins, and while proteins are long spiral molecule chains, there are two main types, fibrous and globular. Fibrous proteins are the major structural material of animal tissue. They're found in things like your hair and your nails and your muscles, the things that your body uses for structure. They have long spiral chains folded to form long thin shapes, and they are strong and generally insoluble in water. And there's a diagram of them to the left there where they look very long, although they are coiled round one another. Another type of protein is a globular protein. And globular proteins are involved in the regulation of life processes. For example, hemoglobin, which is used to transport oxygen around the body, and certain hormones like insulin and enzymes, which are used to regulate different chemical reactions in the body, are all examples of globular proteins. And they have spiral chains which are folded into spherical shapes, and are generally soluble in water. So fibrous proteins are insoluble in water, globular proteins are usually soluble in water. So proteins can be folded into these shapes because they're able to hydrogen bond. So remember that proteins have a peptide link made up of an NH and a C double bond O. The NH can hydrogen bond, and those hydrogen bonds can differentiate between the structures of proteins. So some proteins will be held together by hydrogen bonds, making them globular. Some proteins will be held together by hydrogen bonds, making them fibrous. So proteins are able to hydrogen bond to themselves and other proteins through their polar CO and NH groups. This means that the hydrogen bonds between H, N, O or F occur readily, and this causes proteins to produce complex structures such as the alpha helix and the beta pleated sheet. Hemoglobin, as shown, also has very complex structures and specific shapes which help it carry oxygen around the body. A very important use of proteins in the body is actually enzymes, and we will have heard about enzymes before, especially if you do biology, you've heard a lot about enzymes. Enzymes are proteins which act as biological catalysts, and catalysts are things which speed up chemical reactions. So enzymes are used to speed up chemical reactions in the body. In the egg white, the protein is an example of a globular protein molecule, like a ball of string that's all bonded together by hydrogen bonds to keep it in the ball shape. This structure is held together by hydrogen bonds, and when heat is applied, this heat energy breaks the hydrogen bonds between protein chains, causing the long chains to unfold. So when you apply heat to an enzyme, for example, the heat causes the hydrogen bonds to break apart, which in turn renders the enzyme not useful, or it can't be used to speed up those chemical reactions. And that we call that denatured when we break these hydrogen bonds in between molecules. So due to the protein chains now being uncurled, they can then form intermolecular bonds with other uncurled molecules, changing the shape completely. This process is known as denaturing. It also occurs when we expose enzymes to higher temperatures. It no longer has the specific shape that allows it to interact with the reactant molecules. So when you take an enzyme and you heat it up, there's two things that happen to the enzyme. It becomes denatured, and by becoming denatured, it actually changes shape. In your pupil booklets, there are a couple of questions that we are going to go through and answer. So please try these questions on your own, and then you can go through the solutions with me. So the first question says, draw the protein formed from the reaction of the following amino acids. So remember that reacting amino acids together is a condensation reaction where we are losing water for every peptide link that we make. So I'll be losing the OH of this molecule and the H of this molecule to make a link between this carbon and this nitrogen. Likewise, I'll be making a new link between this carbon and this nitrogen, and in doing so, I will lose this OH and this H. So the hydroxyl group, the OH always comes from, is always lost from the carboxylic acid, and the H is always lost from the amino group. And now I can draw my protein out without the highlighted OH and H's because they'll be lost as water molecules, but with new bonds where the dotted bonds are on my diagram above. So we'll start off and we'll draw it from left to right. So we've got an N with two H's attached, carbon with our R group and our hydrogen, our C double bond O. I've lost this group here, so I'm not going to draw this in, but my dotted line is now a new bond between carbon and nitrogen. I've lost this, so I'm not going to draw this in, but there's still this hydrogen here which is left. We then have a carbon joined to a CH2 group and then a hydroxyl group. Our C double bond O, and I've lost this OH, so I'm not drawing that in, but I am drawing in my new bond, which is a dotted line up here. NH, I've lost this H, I'm not drawing that in. Carbon, CH3, C double bond O, OH. So that is my new protein molecule. And I can see that my protein molecule is made up of 3 amino acids, so technically this is a tripeptide. And my peptide links are here, C double bond O NH, C double bond O NH. 2 new peptide links in my protein. Question 2 says, draw the amino acid that would be produced from the following protein if it was broken down. So, if we were to break this protein down, first of all I need to identify the peptide link. And my peptide link would be here, C double bond O NH, and that is the only peptide link in the molecule. So breaking this protein down, I would be breaking the bond between the carbon and the nitrogen of that amide link. And that would produce 2 amino acids. This is the first amino acid, this is the second amino acid. So all I'm doing is drawing those amino acids out, but I need to remember to add on the hydrogen and the OH, which is going to be added back in from the water. This is a hydrolysis reaction where we add water back in. So I'd be adding water here, plus water, to split that molecule up. So my first amino acid, I'm going to start it from the right hand side. I'm copying it out exactly as they have got it there. I'm copying it out exactly as they have it here. H2NCH, CH3 and C double bond O. And I will also get an OH added onto the end here from the water that I'm adding back in. That is my first amino acid, this here. My second amino acid would be H, CH, H. So I'm now drawing this amino acid here. C double bond O, OH. And again, we need to remember that a hydrogen would also be added on to that side when it's broken from the water you're putting in to break it up. So either one of these two would be acceptable as long as they are completed with the hydrogen and the OH group that we had to add back in from the water. Question 2b. So part B says state the name of the reaction that breaks down proteins. The name of the reaction is hydrolysis. Question 3 says how many amino acids make up this protein? Well, first of all, we need to identify the peptide links. That's one peptide link, C double bond O NH, C double bond O NH. So there's two peptide links here, and if I was to break those peptide links between the carbon and the nitrogen, I would make one amino acid, this would be a second amino acid, and this would be a third amino acid. 1, 2, 3. So there's 3 amino acids there. And question B says draw any of the amino acids present. So 3b. Now if we start with amino acid 1, we would have NH, or amino group. I have added a hydrogen on here to complete the amino group. This part of it I'm just copying from the diagram that they have given. CH2, CH3, and a C double bond O, and to complete this I would have to add in our OH onto that carboxyl group. Another alternative is amino acid number 2. Again, I've got NH and I'm having to add on an H at the end here to complete the amino functional group of the amino acid. And then CH2, 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 NH2. And C double bond O. And again, I'm adding on an OH there to complete the amino acid. Or the final one, hopefully I can squeeze it in here, is amino acid 3. NH, again I'm adding in an H to complete the amino group. CH, C double bond O, and I would need to add in the OH to complete the carboxyl group of the amino acid. And we have this ring down the bottom as well. C, N, C, double bond N, C. Just making sure I add in all the hydrogens where they should be. So that is our 3 different amino acids, amino acid 1, amino acid 2 and amino acid 3. The only thing you have to remember to do is complete the functional groups on the amino acids by adding in hydrogens to the amino groups and hydroxyls to the carboxylic acid groups. So I have added in all of these to complete the functional groups.